Okay.
Thank you, Chris. So, good afternoon, everybody. My name is Chris. I'm from Hancock College, chemical engineering student, and my mentor under the tutelage of Charles Hijema and the O'Malley Lab with Professor Michelle O'Malley. Thank you for making it today. So the research in our lab is primarily studying anaerobic fungi for use in biofuel applications. So why would we want to, take part in this research anyways? Well, anaerobic fungi are found in the guts of ruminant herbivores, so they're thought to be the primary degraders of plant biomass in all these animals. So why would we want to go through all this trouble and dig through horse poop and isolate these fungi and deal with all the unpleasant smells in the lab to do this research? But it's because anaerobic fungi, they naturally break down cellulose in plants into sugars. So they do it very efficiently, and the breakdown followed by the fermentation of these sugars would lead to valuable products. So this is like a model of like what would be in like a plant. It's a very rigid structure, and so if we could break that down into glucose and ferment it into some kind of valuable product. But why do we care? It's just because they're a promising renewable energy source. It would provide us with something that has a net positive energy output, unlike petroleum, and it would help strengthen the economy, lower our dependence on foreign oil, and there's a whole bunch of other applications to it. I don't want to specifically emphasize on just the fuel, but you can make pretty much anything out of just the fermentation of sugar. But the huge bottleneck right now is the breakdown. It's very inefficient, and we would like to solve the problem of how to break them down efficiently. So right now, there's not a lot of information on the structures of these anaerobic fungi and exactly how they break down cellulose into sugars. So these right here are the enzymes, the little Pac-Man looking guys. And then these two are just proteins, sort of like a little lock and key mechanism, just for the general structure of the enzymes. These little hexagonal rigid structures represent the plant polymers. And here are two models basically of what we hypothesize they look like. This one is more of them anchored around a scaffolding, and this one, the enzymes more flow freely throughout the structure of the plant. So, and they're anchored upon themselves. So what is known about them so far is we know that they assemble into large complexes, and they break down plant mass in mechanical and chemical through those processes. And we also know the composition of these Docker domains. So you can think of like cellulase as like the boat, and then the dock, and then the harbor, and then the land. So and more of like a simple way of putting it. And so what is unknown is these guys, these cohesion binding partners, and also which of these models is more accurate. So this is what I have been doing over the last 8 weeks, is trying to identify what this is composed of. So I want to identify these, and also was pretty ambitious into identifying which of those models is more correct. So an intermediate goal of this research was just understanding how they work completely, basically, and then a long-term goal would be this. And so the methods that we've been using, we've been trying to do cross-linking experiments to see how these Dockerin proteins interact and try to identify their binding partners. So this looks like a pretty complicated procedure, but I kind of like to think of it as like getting introduced to like your girlfriend through a mutual friend. So, so protein 1's like, oh, I'm gonna go to the party and like I'm really nervous. Protein 2 is gonna be there. I don't know what to say. So here comes the cross-linking reagent, and together they bind to each other, and then the second step of the reaction they get introduced, and then off goes the other guy and these guys. And then we observe how these two interact. You know what I mean? So before the cross-linking can be executed, we need pure protein, and that sounds like a pretty simple thing to do, but it's definitely not. It's been pretty much the main thing that I've been trying to do during this whole thing, is just trying to get pure protein from a bunch of different doctrines. So basically the procedure takes a few days. We start the culture on day 1. We just take cells and grow them in a test tube, frozen cells, and then upscale them overnight on the second day, and then collect, break open the cells in a centrifuge, and we use little silica beads to break them open. Collect the soluble fraction, the proteins will be then dissolved in the solution. Run them through a nickel column. I'll have a diagram on the next page, and then we check them for purity by running them through a gel, and it separates the ions according to mass. And I'll have a little diagram later. So just to summarize that, we start a culture, break open the cells, and run them through a nickel column. So shout out to all my Dragon Ball Z fans in the crowd. So, and here's a little diagram of the nickel column. So our desired protein, it flows through the column and binds to these nickel ions, while all the other ones, the impurities, are just sort of floating around in the solution, and then we run it through what's called an loop. It needs to break away. So once all those impurities are out, we break off our desired protein from the nickel, and there we go, we have pure protein, theoretically. So the results that I've come up with, finally we have a nice sample to be used in our cross-linking experiment. So the units here are kilodaltons, basically like an AMU. And so here are our proteins of interest that we've identified. So the key thing is like we have one nice defined line. That's how we know we have nice pure protein. So this one was a success, and these are the ones that we actually used in cross-linking experiments to identify binding partners for these proteins. So what we did, we ran it through a cross-linking experiment, and then we run it through the gel again, and try to identify what it's binding to, the relative mass of what it is. And so this right here we found was our cross-linking, or just the general mass of what our cross-link protein is binding to. So, but still, we have an idea of how much it weighs, but we still don't know exactly what it is. So further research would include sending that off to mass spec and actually finding the actual composition of the protein. So that was a success, I mean, for me at least. So as a summary, we have identified a binding partner for our protein using a chemical cross-linker. And so further research that we might want to do is send this off to mass spec, as I said. Still continue to characterize the gut fungal enzyme complex, and use this to actually engineer our own microbes to turn what may be trash into free money. So special thanks to everybody in the INSET program, Jens, Leanne, Maria, all my interns, fellow interns that have made the experience very pleasurable for me, Dr. Nick, and to Charles and Kevin, and thank Charles for being very patient with me. I know as much of a task it is for me to learn all this stuff, it's probably just an equally difficult task to teach me all this stuff. And a man that does not get nearly as much credit as he deserves, Mr. Don Del Bello. He would not be here today without him, so thank you.